Welcome to the Proteomics course. Today's lecture is on proteomics and systems biology. The lecture outline: first I will start on proteomics and then we will move on to the systems biology. Today we will continue on first proteomics and then how different type of omic data can be applied for systems approach analysis, that will be discussed. So, in this slide I have shown you how you can define different type of omics platform. So, omics is a suffix derived from the Greek word om, means all or every. The omics is used as a suffix which has enabled the explosion of terms genomics, transcriptomics, proteomics and metabolomics and so on and so forth. The omics also implies an integration of biology with information science and conveys large scale biology using systems approach. So, as you can see the slide, if you are studying about DNA, that will be in totality known as genomics, RNA study and transcriptomics, proteins in proteomics and metabolites in metabolomics. If you are looking at all of the cellular contents of proteome, that will be known as cellular proteomics or cellular genomics, and similarly all of the proteome of an organism will be known as global proteomics, or similarly at the gene level global genomics. So, let us first start with proteomics. So, if you remember from the previous lecture, proteome is set of all the proteins which are expressed by a genome. Proteomics is a study of proteins and their properties to provide an integrated view of cellular processes. What are these different properties? These properties include the extent of the protein expression, how different type of post translational modification and co-modifications occurs in the cell, different type of enzyme regulation, whether it is activation or inactivation, and then different type of intermolecular protein-protein interactions. The current goals of proteomics are very broad. It is including the diverse properties of proteins, which we have discussed in the earlier lectures, looking at the vast side chain chemistry of amino acids and different levels of protein structures. So, whether it is sequence, quantity, the state of modification, activity, interactions of proteins with other proteins and other biomolecules, and subcellular distributions and structure analysis, all of these are broad goals of proteomics. Now, central dogma concept was discussed earlier, which is an orderly and unidirectional flow of information encoded in base sequences of cells passed on from DNA to RNA and then to the proteins. This is the simplest definition of central dogma. The genome sequencing projects, they have provided researchers with an unprecedented information of genome sequences. However, there are numerous proteins which can be encoded by the genome. Therefore, analysis of the static genome by doing sequencing alone is not sufficient. A gene can code for several type of proteins because of its alternative splicing and post transcriptional and post translational modifications. Therefore, it suggests that studying proteins is more challenging than genome or transcriptome. Therefore, proteomics has great significance to understand the biological systems. The completion of genome sequencing projects of several organisms, including human, has been one of the most remarkable achievements of this century. However, these have not been sufficient to unravel the mystery of complex biological processes. The similar gene numbers of many diverse group of organisms has failed to explain their varying biological complexity. A more meaningful understanding of biological function can be obtained through the characterization of products of gene expression, the protein, which serve as ultimate effector molecule of biological systems. The proteomics refer to the study of entire protein complement expressed by an organism at any given time. While the genome of an organism is mostly static, the proteome is dynamic and it changes with environment and time, thereby elevating its complexity level. The gene regulation is regulated by several post-transcriptional and post-translational modifications, due to which the number of proteins expressed in a cell is much greater than its genomic counterpart. The proteomics aims to decipher the structure and function of all proteins in a given cell under specific conditions and to obtain a global view of cellular processes at the protein level. Study at the DNA level known as genomics, RNA level transcriptomics and protein level proteomics. Analysis of the proteome involves protein extraction, separation, identification and finally, characterization of various proteins. The various proteomic techniques which are currently employed in different applications. We will talk in detail about different proteomic technologies in the subsequent modules of this course, but briefly, very broadly you can group these technologies as gel based methods, gel free mass spectrometry based methods, mass spectrometry techniques, techniques to study the protein interactions and structural proteomics. As I have shown some abbreviation in this slide, broadly different type of gel based proteomics such as sodium dodecyl sulfate, polyacrylamide gel electrophoresis, SDS page, two-dimensional electrophoresis 2DE, difference in gel electrophoresis DIGE, blue native page, as well as different type of staining methods such as Kumasi, silver, fluorescent dyes and phosphor stains such as procodiamond and multiplex staining methods. All these can be grouped under gel based methods. The gel free methods, especially in the mass spectrometry side, includes SILAC, which is stable isotope labeling by amino acids in cell culture, CDIT, culture derived isotope tags, ICAT, isotope coded affinity tagging, VICAT, visible isotope coded affinity tagging, MCAT is mass coded affinity tagging, and then QUEST, which is quantitation using enhanced signal tags, ITRAC, isobaric tagging for relative and absolute quantitation, GIST, global internal standard technology, ICPL, isotope coded protein labeling, AQUA, absolute quantitation, CISCAPA, stable isotope standards and capture by antipeptide antibodies, COFRADIC, combined fractional diagonal chromatography, and MUTPIT, which is multi-dimensional protein identification technology. All of these are various new advancements in the gel free methodologies. The basic mass spectrometry, which is central to the proteomic application, includes different type of ionization sources such as matrix assisted laser desorption ionization MALDI, electrospray ionization ESI, and different type of mass analyzers such as quadrupole, time of flight, ion trap, and Fourier transform mass spectrometry. Then different type of tandem MS based systems are also used. The surface enhanced laser desorption ionization time of flight CELDITOF is also used for various clinical applications. The protein interaction methodologies include immunoprecipitation, yeast to hybrid methods, and different type of protein microarray platforms such as antibody arrays, nucleic acid programmable protein arrays, multiple spotting techniques, and various other cell based and cell free expression based protein microarrays. The detection can be either based on the labeled methods using fluorescence, chemiluminescence or radioactivity, or it could be different type of label free methods such as surface plasmon resonance, interferometry based methods, or different type of conductance based methods employing nanotubes and nanowires. The structural proteomics, it involves X-ray crystallography, nuclear magnetic resonance NMR, transverse relaxation optimization spectroscopy TROSI, circular dichroism CD, and different type of microscopy methods including atomic force microscopy and electron microscopy. So, as we have seen, there are large number of proteomic technologies which are currently available for various applications. Many times, to address one biological question, different type of methodologies come together and then provide solutions to that problem. For example, looking at some clinical sample for identification of biomarkers of a disease, one can employ the samples such as tissue or blood or different type of body fluids, and then either directly extract the protein and subject to the mass spectrometry, or first resolve on two dimensional electrophoresis followed by identification on mass spectrometry, or take these samples directly apply on the microarray based platforms and then detect using label based or label free methodologies. Eventually, these type of results will enhance knowledge for the monitoring the therapy response as well as identification of early disease diagnosis. This is just one example. Similarly, multiple type of proteomic technologies can be used for different applications. There are several proteomic techniques which are employed for studying these proteins, such as two-dimensional gel electrophoresis, mass spectrometry, protein microarrays, as well as some label free detection techniques such as surface plasmon resonance SPR. After discussing proteomics, now let us talk about systems biology. So, what is systems biology? Is that an historic knowledge, a method to understand biological systems, or a tool to solve the practical problems? Systems biology is the examination of a biological entity as an integrated system rather than the study of its individual characteristic reactions and components, which is termed as systems biology. A study of all the mechanisms underlying the complex biological processes in the form of integrated system of many interacting components is studied under systems biology. The systems level understanding of biological networks require information from different levels. As you can see, from DNA to RNA to proteins forming systems, and then that information can be applied to understand a complex system for different organisms. The biological information is represented by the networks of interacting elements and dynamic responses to the perturbations. These networks provide insights which cannot be analyzed from the isolated components of the system. The common elements of the systems biology include networks, modeling, computation and dynamic properties. The different type of biological networks such as protein-protein interaction networks, gene regulatory networks, protein and DNA interaction networks, protein lipid, protein other biomolecules network and metabolic networks. The various ingredients of systems biology. For example, if you are studying about a cell and the system's behavior, one need to look at the genome, its transcriptome profile, proteome profile, how protein, DNA and different type of transcriptional networks are altered, protein-protein signaling networks, multimeric complexes, how they are formed, how protein is localized in the intracellular dynamics and metabolic networks. So, all of these are ingredients of studying about a system. Now, systems biology study can be done at different levels. For example, to study the complex physiology of human, one can look at individual systems such as respiratory systems, nervous system or other physiological systems. Studies can be done at the intercellular or intracellular level and finally, at the molecular level involving genomics, transcriptomics and proteomics. So, why there is need for systems biology? The study of biology at the system and subsystem level for understanding the biological processes and network is very much required. As you can see, to understand even simplest system of a cell, how it is regulated with its extracellular space, the cytoplasm and different other components. Examination of the structure and dynamics of cellular and organismal function is very much required for understanding the systems, rather than the characteristics of isolated parts of the cell or organism. So, what is the aim of systems biology? To understand the biology in holistic approach rather than the reductionist approach. The systems biology aims to quantitate the qualitative biological data and provide some level of predictions by applying different type of computational modeling. The systems biology approach involves first of all collection of large experimental data sets and then mathematical models to provide insight of some significant aspects of the data sets. A simple systems biology approach would involve experiments by adding new data sets which will be used for model constructions and model analysis, and the biological insight derived from these models can be used to propose new hypotheses. So, the properties of a systems are probably more than just the sum of all its individual properties or its components. Therefore, it is possible that system may have its own property by applying all the components. So, what are different approaches have been taken to study the systems biology? The distinct approaches of systems biology include the model based and data based methods. The model based approach involves some prior information which can be implemented in the models, whereas in the database objective is to find the new phenomenon. The model based relies on computational modeling and simulation tools, whereas the database method relies on the omics datasets. So, in model based systems biology approach, it is difficult to build the detailed kinetic models, but in database system, the complex relationship among the various type of omics information and metabolic pathways and networks can be created. Studying systems component is very challenging. Systems biology and biological network modeling aims to understand the system's structure and function for better understanding of system properties like its robustness, as well as use for the prediction of systems behavior in response to the perturbations. The reductionist approach involves disintegrating the system into its component parts and studying them, whereas the integrative approach involves integrating the study of individual components to form conclusions about the system. What is systems biology triangle? So, first of all, the system's information is generated at various levels. As we have discussed, starting from genes to mRNA to proteins to metabolite, or identifying regulatory motifs, metabolic pathways, functional modules and different large scale organizations. This information has to be stored, processed and further executed to identify the system level information. Even simplest system such as cell can be linked with various properties. Its genome sequences of different molecules, intracellular signals, transcription factors, different type of cis binding activities, the expression profiling of RNA and proteins and different type of cellular processes. So, what approach one can take to study about the system? Extraction and mining of complex and quantitative biological data, integration and analysis of these data sets for the development of mechanistic, mathematical and computational models, validation of these models by retesting and refining after proposing some hypothesis. Different online databases and repositories are nowadays developed for sharing large data sets and various systems models. The systematic approach to study how molecules act together within the network of interaction that make up life is definitely going to be useful to understand the systems biology. The systems biology triangle, as you can see here, involves the experimental data set could be derived from different type of omics platforms, technologies, how the computational analysis can be performed, different type of bioinformatics softwares and tools, and then computational modelings by obtaining some theoretical concepts. The synergistic application of the experiment, theory and technology with modeling to enhance the understanding of biological processes as a whole system rather than the isolated part is termed as systems biology triangle. So, the systems biology triangle, the wet lab experiments or bioinformatics based data analysis can be used to propose a model. The model building as an aid to understand the complex system and some hypothesis can be generated which could be used further to propose more quantitative models or predictive models, and also it can be used for independent techniques for the model validation. So, what is system study? First of all, the difference between systems study and the component study one need to understand, and this is what we have tried to emphasize in the previous slides. After generating the data set and creating the systems biology triangle, then this information can be used for understanding the systems in the more complex and mechanistic level. Systems study and model building. The system science includes synthesis, modeling concepts, analysis. Life sciences discipline provides quantitative measurements, genetic modifications, and deriving some hypotheses. The information sciences enables the visualization, the modeling tools, and different databases. So, this model building as an aid to understand the complex system is very useful for systems level investigation. System is an entity which maintains its existence through a mutual interaction of its constituent parts. The systems biology research consists of identification of the parts, characterization of the components, exclude the ones which are not a part of the system, identify the interaction of the components with each other, and identify the interaction of the components with the environment which modulate the parts either directly or indirectly through modulation of internal interactions. The systems biology concept can be understood with help of two approaches such as reductionist approach and integrative approach. The reductionist approach focuses on disintegrating the system into its component parts and studying them, whereas the integrative approach focuses on integrating the study of individual component to form conclusions about the system. Consider a cell with its component molecules. Let us say we want to study the metabolic pathway as a biological system. When the environment of the cell is perturbed a little, the individual components undergo unique changes such as increase in the production rate or decrease in their amount. At this stage, due to lack of knowledge of the nature of interaction of proteins, we cannot interpret how the system gets affected. But when we study the interaction of one component with the other, we can conclude that the increase in rate if DNA binding protein leads to increase in the synthesized amount of DNA, which further changes the final amount of lipoproteins produced. Thus, we can see that to study a system, we need to analyze not just the components, but their interactions. These biological systems can be protein-protein interaction networks, gene regulatory networks, protein DNA networks, protein lipid networks, and metabolic networks. To study the system, we need to know about the components and its interactions. The data about the components comes from genomic and proteomic studies. The information about the molecular interactions comes from the interactomic studies. Here it is shown that in a systems approach, experiment, technology and computational modeling, this triangle is very important, which has to be linked with the theory to form a systems triangle. Now, let us try to integrate the omics approaches with systems biology. So, genome sequencing projects in genomics era from 1990s to 2000 accelerated the pace of omics research. Then from 2000 onwards, proteomics field also got accelerated and new methodologies, new tools came into the place for studying the proteome, and the data derived from genomics, transcriptomics, proteomics, metabolomics and other omics approaches have now brought the integration of these data sets in the systems biology field. The system study requires obtaining data sets from different approaches and analyzing them. For example, as shown in the slide, the genome wide data sets can be derived at the genome level and looking at the expression of the different transcripts, or at the proteome level looking at different type of protein interactions. These data sets can be stored in the clinical databases, and also it can be mined from the literature manual curation. Then integration of the orthogonal data sets further can be used for validating the networks and deriving, identifying therapeutic targets. Further, it can be used for experimental validation. Studying systems cannot be done in isolation in individual labs. It requires different expertise and collaborations from scientists from different disciplines of biology, physics, engineering, chemistry, computer science, mathematics, medicine, statistics and many more. So, eventual aim of this goal of this current systems biology field is to employ the omics level information obtained from different levels from genome, transcriptome and proteome, derive that information at the systems level, integrate, quantitate some models and then propose and use it for the understanding the physiology and apply that in medicine. So, this omics to physiology, this flow can be well maintained by employing systems biology tools. What are the challenges of systems biology? Systems biology is extremely challenging. The emphasis is to understand a system. Understanding dynamics of even simplest biological networks not only requires only the understanding of biology, but also its modeling and simulation. The disintegrative study can be used for studying from cells to proteins to gene, or integrative study could be used for putting these pieces back together again and then understanding and doing the prediction and control of functional biological processes. So, all of these are very challenging, but currently being addressed by applying various systems level tools. So, how proteomics and systems biology are integrated? Proteomics, as we have studied, it is useful to understand the complex signaling networks in biological systems. It is very indispensable tool for systems biology. The global analysis of proteome is important. However, there are many limitations. In each experiment, only thousands of proteins can be studied. Therefore, new approaches and systems level investigation and predictions are required. The system investigation is required to study the complex dynamic structure interaction with the biological systems, whether it is at cellular level or at the organism level, and ultimately it is responsible for their function and behavior. So, in summary, today we discussed that how omics era, the technological advancement in genomics, proteomics and metabolomics have generated large scale data sets in all the aspects of biology. These large data sets has motivated the computational biologist and systems approaches with objective of understanding the biological system as a whole. While proteomics continues to generate the quality data at the proteome level, the systems biology approach characterizes and predicts these dynamic properties of biological networks. Now, in the next module, we will focus in more detail different type of proteomic technologies. Thank you.